We're picking up with regard to the last part of the laboratory assessment, and one of the things that we often will do is request a blood, in particular an analysis which is a CBC and a differential. And what the differential does is basically give us a breakdown of the different cells that are there. That can give us an idea whether there's a problem with an anemia by looking at the red blood cells. It will tell us if there's a problem with platelets and clotting by looking at the platelets, and then by looking at the white blood cells, it'll give us an idea whether there's an infection. So one of the other things that we will get when we're doing the blood is a hematocrit, which is simply, it's sometimes referred to as a packed cell volume, and that represents the percent of red blood cells. Basically, a blood sample is taken, spun down, and the red blood cells, white blood cells, the solid portion of the blood gets spun down to the bottom portion of the tube, and basically the liquid part of the blood stays to the top. So it's really giving us a percentage of red blood cells. If the hematocrit is low, this may be due to a reduction in either the number of red blood cells or even the size of the red blood cells. Or another thing that may happen is sometimes the plasma volume may increase. So this can give us an assessment, sometimes indirectly, of the red blood cell function there. The red blood cell count will tell us how many red blood cells are there. It will tell us if we have an anemia. It's also an indirect estimate of how much hemoglobin will be there. So that becomes an important parameter we need for establishing whether the person has an anemia. Slide 48 is looking at the white blood cells, or is describing those. When we see that the white blood cell count is elevated, and they describe it as a shift to the left, that basically means that there is the potential for either an infection or inflammation or sometimes trauma, but generally it's associated with infection or inflammation. If there is a reduction in white blood cells, this can be due to a reduction in specific white blood cells, such as, for example, the neutrophils are low, that would be a neutropenia. If there's all of the different white blood cells, that would be referred to as a granulocytopenia, and that's what occurs in an aplastic anemia. So a number of drugs may attack the bone marrow and may actually produce reductions in white blood cells. This makes the individual susceptible to infections. We also see if there is an eosinophilia, which is representing the eosinophils. Those, we see that type of condition occurring when we have allergic reactions or occasionally with parasites. Slide 49 describes the platelets, and generally we are looking at a thrombocytopenia when there is a reduction in platelets. This is sometimes referred to as thrombocytopenia purpura. It may be referred to or may be caused by what is known as DIC, which is disseminated intravascular coagulation, in which we have the cells or the platelets get tied up in a bunch of little tiny clots that are ineffective, or this may occur as a consequence of development of aplastic anemia. We may also see a thrombocytosis, which may be due to trauma or splenectomy or leukemia. So this would be alterations in the platelet numbers. Generally, one indication that we have a platelet problem is alterations in bleeding. When we have a thrombocytopenia, generally these individuals will have increased bruising. They will have potential nosebleeds, bleeding from the gums, and bleeding can be very severe depending on how many platelets are reduced. Again, this can be determined by looking at a CBC and getting a differential count. Another thing that may happen, I touched on this a few minutes ago on the previous slide set, is that we may have a problem with clotting, and we look at prothrombin times. Generally, that's used to monitor patients that are on Coumadin therapy. Sometimes they will look at a PTR, which is a prothrombin time ratio, but most frequently what will be reported is an INR. And INR stands for International Normalized Ratio, which basically is a means of normalizing prothrombin times and allowing you to compare them across different parts of the country and through different hospitals and so forth. We also will use partial thromboplastin time, another index for clotting, and that one is used to monitor heparin therapy. But mostly you will see for monitoring clotting, we will often see the INR used as the main issue there. Now we're going to shift gears here as we go to slide 51, and I want to pick up on a little bit of basic physiology that builds upon some of these other slides. So remember that on slide 51 is a diagram of the membrane, cell membrane, and want to just reemphasize and remind you that it's a lipid bilayer. We have proteins on the outside and on the very inside, and then the lipid bilayer that is between these two proteins. These lipids, lipophilic drugs can pass through this bilayer. We also have these aqueous pores that are present that also allows substances to go across that. But we have this is the composition of the cell membrane. Again, the lipid bilayer. In slide 52, it just depicts a membrane transport mechanism. Sometimes the way substances, including drugs, may get across a cell is that they may be literally hooked into or attached to a carrier on the inside of the cell. The drug cannot pass through the cell itself because of its properties, but it may attach to a carrier which literally will transport it through the cell. When it gets to the other side of the cell, the carrier will then release it. So we do have these membrane transport mechanisms as depicted in slide 52. Slide 53 is just demonstrating that there's a number of different transport processes that we will talk about as far as how substances move across cells. The first one is a passive transport. What's happening here is literally that it's almost like something is sliding downhill. It's usually going from a gradient that is from high to low, and again does not require energy, and this is a passive transport process. On this diagram in slide 53 you can also see the middle one, which is a facilitated process. It again is traveling down a gradient, but being facilitated, we have some sort of energy that's applied to facilitate or make it easier. So that is called a facilitated. The analogy here would be like a wagon with the wheels helping facilitate it versus having just a sled that's sliding down as if a passive. When we have to go against a gradient, we are going to have what is known as an active transport process, which is going to require energy, and primarily it's going to need, we use ATPases as a form of energy in a lot of these. Now, the next few slides, starting with slide 54, are the criteria for a transmitter. This is something that you should make sure you know. I think most of you have had this before, so I'm going to cover them very quickly. But these are the things that we identify whether a substance is a neurotransmitter. First of all, it has to be present in the neuron. We have to have the necessary things to synthesize it that are present in the neuron, usually enzymes. The precursors and cofactors necessary to make the transmitter have to be present in the neurons. So literally the building blocks have to be there. We find that the release of the substance is calcium dependent, and that requires calcium, and it occurs during depolarization. These criteria continue on the next slide, 55, which we have some sort of mechanism for inactivating it. This may be sometimes an enzyme, it may be sometimes a transport process or a reuptake process. Another important thing is that when we apply the substance to a tissue, it has the same effect as if we stimulate the nerve. And then if we have drugs that or substances that are antagonizing the substance, we would see that the antagonist would interact with the substance and nerve stimulation in the same manner. So in other words, if we stimulate the nerve and we put an antagonist there, it blocks the effect. If we put the proposed transmitter in there, the antagonist would block the effect. Also, what we would see is that there are specific receptors that are present for the transmitter to interact with. And then that is the basic component that we see for criteria for neurotransmitters. Now, the next series of slides are going to review the autonomic nervous system. Very important component that you need to be aware of, and make sure you know these slides because these tend to show up in various ways on PEP exams and so forth. First of all, we're going to talk about the autonomic nervous system, which is going to, as indicated here on slide 56, is going to basically regulate the moment-to-moment activity of pretty much most of your organs. It's an involuntary system, which means you don't have to think to do it. Mostly what you're going to see is the organs are going to be dually innervated. In other words, most organs have sympathetic and parasympathetic innervation, and they generally oppose one another. So on slide 57, and I alluded to this earlier in these presentations, we have the sympathetic nervous system. Where it originates is from the thoracolumbar outflow. This basically serves survival functions, pretty much to prepare the body to react to stressful situations. Often we refer to it as this transmitter or the system that is involved in fight or flight. The transmitters are norepinephrine and epinephrine, with the majority of the nerves releasing norepinephrine. The adrenal gland is going to be involved as a hormonal gland. It will release both norepinephrine and epinephrine. The other system in the autonomic nervous system is the parasympathetic nervous system. It originates from the craniosacral portion of the spinal cord. It tends to be more involved in vegetative functions and maintaining homeostasis. And the transmitter in this case is going to be acetylcholine. So a lot of times this system is called the cholinergic system. Now slide 58 and a few of the subsequent slides are really going to talk about how catecholamines are synthesized. Another important series of slides for you to make sure you understand. We start off with the precursor, which is generally tyrosine, and then tyrosine will be converted to dihydroxyphenylalanine, which is known as DOPA. This is the rate-limiting step for catecholamine synthesis. So it doesn't matter if it's norepinephrine, dopamine, epinephrine. The system that's controlling the whole synthesis process is going to be the conversion of tyrosine to dihydroxyphenylalanine, or DOPA. This occurs in the cytoplasm of the cell. If we continue to look at the synthesis of catecholamines, we go to slide 59, and once DOPA is made, it's converted to dopamine pretty rapidly. That is accomplished with an enzyme known as DOPA decarboxylase, which is a very non-specific enzyme, meaning that other substances can interact with it. This interaction occurs in the cytoplasm, and once dopamine is formed, it's very rapidly taken up into storage vesicles. Once inside the vesicles, then the next step takes place where dopamine will be converted to norepinephrine. And the enzyme available or responsible for this reaction is dopamine beta hydroxylase, and that reaction actually occurs in the vesicles. Now, if this is going to be a dopaminergic neuron, then basically there's no dopamine beta hydroxylase. If it's going to be a noradrenergic neuron releasing norepinephrine, then there will be dopamine beta hydroxylase. So we've got dopamine converted to norepinephrine in the vesicles. Slide 60 goes the next step, in which norepinephrine in certain neurons and in the adrenal medulla will be converted to epinephrine. And again, this is occurring in the vesicles. The enzyme responsible is phenylethanolamine N-methyltransferase, which is often called PNMT. And when PNMT is present, it will convert norepi to epi. Now, once we have the formation of the transmitter, let's talk about how it's stored. And this is discussed on slide 61. Basically, in the vesicles, you have three pools of transmitter. You have the bound vesicular pool, which is actually bound to the inside of the vesicle. You have the free vesicular pool, which is floating around inside of the vesicle. And then the third pool is actually outside the vesicle. It refers to the norepinephrine or dopamine or epinephrine that's actually in the cytoplasm, and that's called the free cytoplasmic pool. The free cytoplasmic pool is going to be important because it's going to regulate the synthesis of the catecholamines, because these storage pools fill up from the inside out. So the bound vesicular pool fills first, then you have the free vesicular pool, and when it fills up, then the overflow goes out and forms the free cytoplasmic pool. When the free cytoplasmic pool increases, what's going to happen is that norepinephrine or catecholamine that's in the floating free in the cytoplasm is going to interfere or inhibit tyrosine hydroxylase. And tyrosine hydroxylase is the rate-limiting enzyme that converts tyrosine to dopa. So when it basically is a good mechanism of shutting off synthesis, because it means the vesicles are filled, we have overflow and shut off the synthesis. So let's talk about transmitter release. Now there's two types of transmitter release as shown on slide 62. Exocytotic release is what we refer to sometimes as physiological release. It occurs when the neuron depolarizes and it is calcium dependent. In other words, it requires calcium for it to occur. Non-exocytotic release is often referred to as indirect release or pharmacologically induced release, which means that it normally we describe it as the drug displaces the catecholamines. It is not calcium dependent. So the difference between exocytotic and non-exocytotic, exocytotic is related to a calcium dependent process occurring with depolarization. Non-exocytotic is not occurring with depolarization, and it is non-calcium dependent and is usually the drug-induced release of transmitter. So let's talk about what happens with release. On slide 63, we describe the exocytotic release. During depolarization of the neuron, calcium will enter the cell. What calcium does is when it enters the cell, it will then interact with two proteins within the cell. One is called neurin, which is a protein that's on the inside of the neuronal membrane. The other is called stenin, which is actually on the vesicles. It's the vesicular protein. When the presence of calcium, what's going to happen is that this forms contractile mechanism. It establishes a contractile mechanism which allows the inside of the membrane, neuronal membrane, to pull the vesicle in, causing a deformation, and will cause the release to occur. Once that release occurs, calcium is pumped out of the cell. The lack of calcium allows these contractile mechanisms to release, and therefore they separate. So exocytotic release allows for calcium to come into the cell through depolarization, which then forms or initiates the formation of these neurostenin interactions. On non-exocytotic release, as I've got identified on slide 64, what we see again, it's calcium independent. It is pharmacologically important. It'll occur in the presence of drugs, but does not occur in the normal physiology. And some of the drugs that can cause this non-exocytotic release, cocaine, amphetamines, tyramine, these are three drugs that can actually cause the release of transmitter, not by depolarizing the cell, but by actually displacing the transmitter from the vesicle. So again, calcium independent. Now, what happens once the transmitter is released? Well, with the adrenergic system or the noradrenergic system, as indicated here on slide 65, the majority of the transmitter that's released by the sympathetic nervous system is going to be taken back up and reused through a reuptake mechanism. Okay? A small portion is going to be metabolized by either monoamine oxidase or COMT, and then a very, very small part is going to either diffuse away from the synapse or be bound to extraneuronal proteins. But the major mechanism of terminating action in the sympathetic nervous system is this first bullet point, which is the reuptake mechanism. We have two types of reuptake mechanisms which are depicted on slide 66. Reuptake 1, which is the primary mechanism, it's going to take the transmitter out of the synapse up into the presynaptic neuron, and then that transmitter, most of it's going to be restored and reused, or a small portion of it may be broken down by monoamine oxidase, which is an enzyme that's present within the presynaptic neuron. Reuptake 2 is sort of the backup mechanism. We refer to it extraneuronal because it takes tissues into extraneuronal tissue, or takes catecholamines into the extraneuronal tissue. And any catecholamine that's taken up into a reuptake 2 mechanism is generally going to be acted on by catechol-O-methyltransferase, which is COMT. Just to go over the enzymes that are involved here is on slide 67, monoamine oxidase, that's going to be presynaptically located. It's associated with the mitochondria. We also see that monoamine oxidase is present in the liver, the lungs, the kidneys, and the GI tract for any transmitter that is picked up in the blood or diffuses to the blood or whatever for it to be metabolized. COMT is primarily in postsynaptic tissue, but we also see it located in the liver, lungs, and kidneys. Now, if we look at slide 68, what we basically are seeing is the neuronal or presynaptic neuron. And you can use this as a review of what happens during depolarization. If you follow this through, we begin with depolarization, where the calcium enters the cell. As you follow this mechanism on down, we find that, for example, at step number 3, we get the calcium going in through the channels. Excuse me, that's step number 4. When the calcium will then interact with the neurostinin, as we see and indicated in step 5, that causes the vesicles to release their transmitter. As shown in 6, the transmitter is released into the synapse. It will interact with these receptors. When it binds to the postsynaptic receptors, basically what it's going to do is it's going to bind reversibly. It doesn't stick there permanently. It basically goes there. I usually describe it as it slaps the receptor and then releases, goes and hits another receptor. And this type of reaction allows a small amount of transmitter to interact with a lot of different receptors. These receptors then usually will interact with opening a postsynaptic channel, allowing enzymes or ions to fluctuate. What happens is ultimately the transmitter is going to be picked back up through a reuptake mechanism, stored back in the vesicle and reused. So slide 68 here shows you sort of an overview of what happens during transmitter release. Now, slide 69 gives you the 4 major sympathetic receptors. We have alpha-1 receptors, which tend to be excitatory. Alpha-2 are inhibitory. Beta-1 are excitatory. Beta-2 are inhibitory. So in general, anything that's a sympathetic receptor that has a 1 subscript, alpha-1, beta-1, tend to be excitatory. So for example, alpha-1 receptors cause vasoconstriction. The alpha-2 receptors tend to be inhibitory. They inhibit transmitter release. Beta-1, primarily in the heart, causes increased contractility, increased heart rate. Beta-2 is inhibitory, tends to cause relaxation of the bronchioles and the blood vessels. Now, I've mentioned the adrenal medulla before. Just want to go back and touch on it as shown in slide 70. There's 2 layers of the adrenal gland. There is the outer cortex. The outer cortex, we refer to as the adrenal cortex, will release the glucocorticoids and the sex hormones. The adrenal medulla, which is the inner portion of the gland, is going to be releasing catecholamines. And what we see is that in the adult, the gland will generally secrete, the adrenal medulla will generally release about 85% is epi and only about 15% is norepinephrine. And again, keep in mind that the adrenal gland is going to be activated during stress responses. Let's move to the parasympathetic system. This is slide 71. So let's start off with the transmitter, which is acetylcholine. Acetylcholine is released from all presynaptic neurons, whether it's sympathetic or parasympathetic, but it's also the terminal transmitter in parasympathetic postganglionic nerve terminals. To make it, we start off with choline, which is a dietary amino acid. It's actually transported into the nerve terminal by a sodium-dependent active transport. This is the actual rate-limiting step in acetylcholine synthesis, is the transport of the choline into the nerve terminal. On slide 72, we continue with the synthesis of acetylcholine. What happens is an enzyme, choline acetyltransferase, will convert the choline to acetylcholine, and in doing this conversion, it's going to get its acetyl group from acetyl-CoA. This is going to occur in the cytoplasm. The storage of acetylcholine is much like what we see in the sympathetic nervous system, stored in 3 pools. And basically what happens here is we don't have the free cytoplasmic pool being a termination of synthesis as we see in the sympathetic system. But basically the free cytoplasmic pool, what happens when the vesicular pool empties, it's going to fill the vesicular pool back up. Another reason for this is we don't have a reuptake mechanism of transmitter in the parasympathetic system because what happens is most of the transmitter is going to be eliminated through an enzymatic reaction. And that leads us to slide 74, which is termination of acetylcholine. What's going to happen is we don't have a reuptake mechanism for the intact acetylcholine. What in fact we have is enzymes, acetylcholinesterases, which are distributed all over the body, in addition to specifically in the cholinergic nerves and in the postsynaptic tissues. And what acetylcholinesterase does is it breaks acetylcholine down into choline, acetic acid, and water. And the choline is the important thing because remember, choline was a precursor, so the choline will actually be taken back up into the nerve terminal and be used for synthesis of new transmitter. On slide 75, I've depicted the cholinergic receptors. For most postganglionic nerves, they're going to release acetylcholine, which is going to stimulate muscarinic receptors. The other two places we see cholinergic receptors is in the ganglia. These are nicotinic 1, they're referred to as NG receptors, and these are in both sympathetic and parasympathetic ganglia. And then we have what is known as nicotinic M receptors, which are the cholinergic receptors, nicotinic receptors that are on the skeletal muscle. The old literature referred to the nicotinic receptors as nicotinic 1 and 2. Nicotinic 1 is analogous to the NG receptor, nicotinic 2 is the NM receptor. Now, just a couple of things to finish up on the autonomic nervous system is that, first of all, we know that the sympathetic nervous system is fight or flight. So when a person becomes stressed, the stress reaction primarily involves activation of the sympathetic system and a inhibition of the parasympathetic system. So on slide 76 here, I've given you some examples of stress reactions and the receptors involved. When you get stressed, heart rate goes up, contractile force goes up. That's due to stimulation of the beta-1 receptor in the heart. You get increased sweating. This is due to activation of the glands, the apocrine glands by epinephrine. Your blood vessels constrict, especially your cutaneous blood vessels, the skin blood vessels. This is due to the alpha-1 receptors, which we have a lot of them in those blood vessels. So it primarily constricts. That's why people tend to get pale when they get afraid. Another thing that's going to happen is your skeletal muscle blood vessels are going to relax, vasodilate, which allows more blood flow to go to those skeletal muscles so you can either fight or run away. Slide 77 shows some more of the stress reaction. So we see that your bronchioles dilate. That's due to stimulation of beta-2 receptors. That helps you bring in more oxygen and breathe. Your pupils dilate. That's referred to as mydriasis, and that's due to stimulation of alpha-1 receptors and beta-2 receptors on the muscles in the eye. And what you have is one muscle constricts and the other one relaxes, which causes pupil dilation. Your coronary blood vessels dilate, which allows more blood flow to your heart. That's due to stimulation of beta-2 receptors. And then the other thing that happens is there's a beta receptor response where that you have glycogenolysis, which is basically you're breaking down the glycogen to form sugar, glucose, so you can use it for energy. Slide 78 is just a reminder that the autonomic nervous system is going to be controlled also by higher brain centers. We have input from the cerebral cortex. For example, fear responses can come there. We also have the more basic responses or control of the cardiovascular system based upon the actions of the hypothalamus and the medullary centers. Now, the last two slides are good pictorials of helping you put the autonomic nervous system into perspective. So slide 79 is basically the fight or flight response. It shows a boy being scared by a bear, which seems to become the international symbol. Every textbook you see uses somebody being scared by a bear as activating their response. But basically, if you read the caption here, it says the sympathetic nervous system is going to provide energy for vital function. You're going to get decreased pulmonary secretions and bronchodilation, which will help you breathe and get more oxygen. Heart rate and contractility go up, increasing cardiac output. Arteriolar constriction occurs, which basically shunts blood from places like the digestive tract to the muscles. The heart's blood vessels dilate. This is the things I talked about. Lipids breaking down, we get glucose. All of those basically help you to prepare for this stress reaction. What we find is gut motility and secretions decrease. Urine becomes retained, and you can't fight a bear when you're urinating. That's their little joke. Slide 80 is the opposite, which is the parasympathetic system. And this one I always like. It is the parasympathetic pig. And it says the parasympathetic pig's heart beats sluggishly while its digestive tract hogs the energy. He's drooling because of increased secretions. He needs to breathe oxygen because his bronchioles are constricted. He's defecating, urinating, and then it says he has an erection. You'll have to picture this, as none of these are shown for modesty. And generally, what you can think about is that whatever you experience with a stress response is your sympathetic nervous system, and you can take the opposite in most cases for determining what the parasympathetic system is. So again, this is sort of an overview of the autonomic nervous system, and basically is going to be helpful for you as we move into the next slide sets where we start talking about specific drugs.